I should let you know that today's material will not be on Friday's midterm.
Yo, Mike, you owe me a drink.
Yeah, so our midterm will be on Friday.
Yeah.
I covered some of this last week, but show up to the same classrooms. We will be talking about translation, but not so much post-translational modifications. There will be a review session after class today, led by students. Looked over the material that they're going to be reviewing. It was fantastic. I have posted a second practice midterm. That's up on the course website. And so some of the students in the class have talked to me about issues with regard to like understanding the material and reviewing the course content, but not really being able to do a good job of providing the kinds of answers that we're looking for. And I think for those students, it will be really helpful just to gain some experience looking over the kinds of questions that we ask. They're maybe kind of a different sort of format than the sorts of exam questions you might have seen in an organic chemistry class or certainly intro. And so we've gone ahead and posted a second practice midterm for those of you who want to spend a little bit more time doing that. And then finally, the comprehensive final exam will be on December 18th in the evening. So look forward to that. And we're getting close to the end here. On Thursday, we'll be getting into the last chapter in the book on gene regulation. So just to review what we were talking about last time, we were pretty much wrapping up the conversation about what happens on the ribosome, and as we've been talking about with DNA-templated DNA polymerization, DNA-templated RNA polymerization, and now RNA-templated protein polymerization, we care a lot about how the cell gets it right. And we really focused on the activity of the amino-acyl tRNA synthetases and their critical role in establishing that fidelity and why we call them the second part of the genetic code. Also, we need to know what's happening at the A site, P site, and E site in the ribosome, know what's going on there. Be able to sketch out the bond-forming chemistry that's occurring on the ribosome. Understand what this phrase means, that the ribosome is a ribozyme, and think about initiation, elongation, and termination processes for these translation events. All right, before I get into today's material, are there any questions about review stuff or material that will be on the midterm? All right. So today we're going to be talking about post-translational modifications. And this slide and the next slide really provides you with the kind of overview that you need. There's two ways to give you that overview. One is just to go through and talk about the different chemical modifications that have been made. Yes, sir?
Do we have to know the nucleotides for the midterm, like memorize them, or will you give them to us?
I think it would be helpful. I hesitate to tell you don't learn them, but I think in a lot of cases, the questions that we'll ask will provide you with nucleobase structures. You should absolutely know the structure of the backbone, the phosphodiester groups, the ribose 5' 3' position, and how the backbone is extended, where the nucleobase is attached onto the ribose. My guess is that we'll probably be giving you nucleobase structures, but you'll be in better shape if you're super familiar with them.
Okay, thank you.
Sure. Okay, so let's think about the types of chemical modifications that we're going to introduce onto amino acid structures. The cell needs proteins to do a lot of different things, both functionally and structurally. And it is remarkable that the cell accomplishes all that, and proteins do so many things, given the limited alphabet 20 amino acids that are coded for by the genetic code and that can be introduced onto the ribosome. We talked about some weirdos like 21st amino acid or whatever, but that's pretty much it. 20 natural amino acids, it's not going to cut it, right? The cell needs proteins to do more than what can just be accomplished by what the ribosome is able to crank out. So we can curate these structures, and these are the critical chemical modifications. We'll really be focusing a lot today on phosphorylation, not so much that it's necessarily more important than any modifications, but today's episode of Meet the Chemist will really be keying in on phosphorylation. So this is really crucial because it introduces a negative charge group onto amino acids that are normally neutral. Other ways to do that will be to sulfate an amino acid position. We can abrogate a charge, remove a charge. So if we wanted to modify lysine, we could acetylate that position. We'll talk about post-translational modifications, not by just adding a very small group like an acetyl group, but actually adding an entire massive protein structure right onto an amino acid side chain, and we'll take a look at ubiquitination and the role that that plays in controlling protein degradation. Lysine primary amino groups can be converted to secondary, tertiary, or quaternary amines by adding a varying number of methylation sites. Methylation can also be introduced onto arginine guanidinium groups as well. We talked before in the class about modification of protein structures by addition of saccharide groups, glycosylation, and I will review that with you guys again today. And then we will also be taking a look at an example of how protein amino acid side chains can be modestly modified by just adding a hydroxy group, and we'll be taking a look at a hydroxylation reaction. So here are the chemical modifications. Yes?
In the case of ubiquitination, why is it useful for us to think about it as adding a protein to an amino acid rather than just extending the polypeptide chain by one residue?
Here is the backbone of the protein structure. So this is not just adding one residue. This actually creates a branched structure, which we don't really see very often in protein chains. And instead of just adding one amino acid at this position, it's adding 70-something residue protein. So this is not your typical minor decoration. This is plopping a huge protein structure down onto this site. And actually, you can add multiple proteins onto that same position in what's called polyubiquitination, and we'll take a look at that as well. Other questions about these chemical modifications? All right. Here's the other way to think about the overview of this topic, which is understanding the role of these post-translational modifications. What does it allow the cell to do? Again, we'll be keying in on this phosphorylation issue, understanding that sometimes these phosphorylation events provide important markers to either switch enzyme activity on or off, or provide some kind of localization signal telling, let's say, a hormone receptor to go into the nucleus. Modification of proteins in the nucleus can be really important in controlling the organization of DNA. I won't spend so much time talking about this today because it will play a critical role in our discussion of gene regulation that will be beginning on Thursday. Understanding that the physical structure of DNA, how it's organized in the chromatin, is really going to be an important part of whether or not any particular region of DNA gets transcribed. Again, glycoproteins have a variety of different functions. We talked about that quite a bit. We understand that very often glycosylation controls trafficking within the cell or determines whether or not a protein will end up on the cell surface or even excreted. Post-translational modifications can be really important in structural proteins, not just things like enzymes and signaling. Localization of proteins can be really targeted to the cell membrane itself by addition of a lipid group. So this controls where the protein is localized within the cell. And then we'll be taking a look at this structure. This is the machinery that controls protein degradation, the proteasome, towards the end of today's lecture. All right. As you review these chemical modifications, I don't just want you to know, okay, it's phosphorylated or dephosphorylated. Like, you know, maybe the biologist might be comfortable with just painting a P on a protein and be done with it. We're not. We need to know what chemical flavor that P comes in and, you know, exactly how it's conjugated onto an amino acid structure and what enzymes are responsible for doing that. So we're going to key in on this today. We'll probably talk a lot about tyrosine phosphorylation, the fact that this hydroxyl group can be replaced by a phosphate group. And very often when we introduce these chemical modifications, they are reversible, so these can be added or removed. In general, there's two different families of enzymes that are responsible for catalyzing the addition or removal of these groups. And that's kind of interesting. It doesn't need to be that way, right? We could ask the same enzymes to do both steps. But in the case of phosphorylation, we'll be taking a look at kinases for addition of the phosphate groups and phosphatases for removing them. Methylation is typically carried out by methyltransferases. And so you should be able to simply sketch the addition of a methyl group to this primary amino on lysine, converting it to a monomethyl lysine or other variations as we add additional methyl groups. Ubiquitination, we'll talk a little bit about the enzymes that are responsible for doing that. In this case here, ubiquitination, and then acetylation. We will just briefly talk about that with regard to acetylating the N-terminal group on lysine. All right, but we're going to talk a lot today about phosphorylation. And this brings up a really interesting question that was posed to me by a student. So we see a lot of examples of how phosphorylation plays a role in signaling. And just to remind you all about how signals proceed from the exterior of the cell to the interior of the cell, right? Lots of times they end up affecting targets that are kinases, that are responsible for carrying out phosphorylation reactions. But typically, as part of that message, the receptor turns over that signal in the form of a small molecule soluble second messenger, such as an isotopic phosphate or cyclic AMP. And we see these second messengers that we talked about before when we were talking about biosignaling come up over and over again. And the question is asked, like, if there's many different G protein coupled receptors or other cell surface receptors, and many of them end up using the same second messenger, how does the cell know what it's responding to? Right? If there's like dozens of different receptors that end up generating cyclic AMP as a second messenger, how does the cell know what is causing the original signaling, what it ought to be responding to? So we don't really have any great answers to these questions. We're still kind of trying to figure some of this stuff out. But this gives you another kind of image of how this might come up. Like, okay, in this particular case, we've got a G protein coupled receptor. and we're going to activate adenylyl cyclase that's going to take ATP and turn it into this important secondary messenger. But this is a really common second messenger. So how does the cell know what signal is coming in and how to feed into downstream results? Well, I think there's a few different ways to answer that question, and one of them is to realize that sometimes the cell doesn't really care all that much what originally evoked the message, that it turns out that what you want the cell to do in response to that message and many others is something like cell division, cell growth. So yeah, if cyclic AMP is having that kind of result, that's cool, no problem. It will be evoked by a number of different messages. The other is to realize that cyclic AMP shown here, like, okay, it's in the cytosol, it's a small molecule. Does that mean that it is floating freely throughout the cell? Concentrations are elevated, and whatever is going to respond to cyclic AMP is going to be affected pretty much homogeneously throughout the cell? Well, perhaps not. And one of the things that we're learning a lot more about is the fact that some components of these signaling pathways are located at very particular positions within the cell. And so even something like a protein kinase might not just be running around freely. It may be located at a very particular position near the cell surface, maybe near a particular lipid raft, where there is a particular G protein coupled receptor. And we are now beginning to learn a lot more about things like scaffolding proteins, or this particular structure, ACAP, A kinase anchoring protein, that tells this kinase that responds to cyclic AMP, hey, hang out here, this is where we want you to receive this particular signal, and now you can go and carry out your effect on downstream substrates. So it's because some of these components of signaling are coordinated physically within the cell, either by their association with proteins like ACAP or on scaffolding proteins, that we can have some measure of control over what that cyclic AMP is really providing a signal for. But again, we're still trying to figure some of this stuff out. It's a kind of a open topic that we're learning more about now. So I also want to emphasize that the messages, the signals that are received, sometimes are conditional. So the cell is capable of regulating a response only when a series of different messages are being received, and that signals can also be amplified. So that if we have some type of cell surface receptor, we can initially activate one component of, let's say, a phosphorylation cascade, and each of these can in turn activate many other downstream effectors, and that these cascades then can dramatically amplify one small initial signal that's received at the cell surface. But at this point, I want to turn the discussion over to an expert in phosphorylation, and that's Professor Neil Shah, who's very kindly come down from Columbia University, where he's just initiated his own research program. And so I'm going to switch the slides over to those that describe his concepts about protein phosphorylation and their role in what happens as far as structure and signaling within the cell. So please welcome Professor Shah.
Thanks for letting me crash your course. So part of the reason I'm here is because I'm deeply interested in protein phosphorylation from the standpoint of protein structure and also the biology that's a consequence of this post-translational modification. But the sort of second half of the reason I'm here is actually because a little over a decade ago, I was literally here. So I actually was an undergrad at NYU, basically in your position, probably 12 years ago or something like that. And I had the good fortune to take a course like this one, and to also work in Professor Kirschenbaum's lab as an undergraduate researcher doing bioorganic chemistry. These two things really got me enticed to move into really understanding how proteins work and how we can make them and manipulate them. So I just moved sort of a stone's throw away uptown to graduate school, where I studied protein chemistry. I couldn't get enough of proteins, as you'll see from the next few slides. And so then I went to study the enzymes that catalyze protein phosphorylation from a structural biology standpoint, understanding how the structures dictate their functions. I did that at UC Berkeley, and then I was fortunate enough to come back to New York City, and just a few months ago I started my own lab up at Columbia University, where we're integrating a bunch of different approaches, chemical, biological, to really understand protein phosphorylation to another level. So that was just to give you a sense of why I'm here. And as Professor Kirschenbaum said, I am here to talk about protein phosphorylation. And you've seen a little bit of the background already from his slides, but just to reiterate, this is a process mediated by a class of enzymes called protein kinases, which uses their substrates, the cofactor or the substrate ATP, which is a phosphoryl donor, and typically phosphorylate the amino acids found in proteins that have hydroxyl groups, such as serine, guanine, or tyrosine. And when a protein kinase brings these two substrates together, this results in the generation of the phosphoamino acid in the context of a protein, so the phosphoprotein. And it's important to note that although protein kinases can actually catalyze the reverse reaction to put the phosphoryl group back on ATP, this is not typically how this is mediated in a cell. Instead, a class of enzymes called protein phosphatases catalyze the removal of this modification through a hydrolysis reaction, basically generating inorganic phosphate and just liberating this amino acid back to its hydroxyl form. So when we think about protein phosphorylation, we tend to, especially in textbooks, focus on the role of these three different amino acids in their phosphorylated states. But in fact, in cells, many different amino acids can be phosphorylated, not just those that have a hydroxyl group. So although the hydroxyl-containing amino acids are phosphorylated most commonly in mammalian cells and other eukaryotic cells, if you look in bacteria, the common currency of exchanging information between proteins in the cell is actually histidine phosphorylation through this weird phosphorus-nitrogen bond. And histidine phosphorylation happens in a relay that happens alongside aspartate phosphorylation, another kind of bizarre-looking molecule. In our own cells, we also see cysteine phosphorylation, lysine phosphorylation, arginine phosphorylation. These are sort of understudied modifications and perhaps less prevalent, but still worth noting. And there are specialized families of kinases that catalyze each of these modifications. Today, though, I'm actually going to focus on one of these sort of more popular ones, phosphotyrosine. Although what I want to stress is that the concepts that I want to illustrate to you about the role of protein phosphorylation can really be translated to understanding phosphorylation of these other amino acids in the context of proteins as well. So from the standpoint of relaying information in a cell, or basically carrying out biosignaling or cell signaling, as we often call it, phosphorylation is a particularly clever strategy for nature to have evolved, for the simple reason that a phosphate introduced onto an amino acid side chain has a dramatic impact on the structure of that amino acid. In the case of serine, for example, it basically doubles the size of the side chain, and perhaps more to the point, it introduces a modification that has two negative charges. So having two negative charges in close proximity creates basically a chemical signature or beacon that's not present naturally or natively rather on proteins as they come off the ribosome. And so because you created this high density of negative charge on a specific site on a protein, this basically creates something you can imagine it being sort of like a beacon for recognition by other proteins. And in fact, what's known is that for each of these commonly phosphorylated amino acids, there are actually specialized protein domains that are found in nature that can specifically bind to just the phosphorylated form. And so to give you one example, there's a class of domains found in all eukaryotes, or rather all animals, I should say, called SH2 domains. And so what these SH2 domains are they're just these 100 amino acid modules that are embedded within other proteins, and they have this deep groove basically on their surface. And this groove is lined with positively charged amino acids. There's an arginine over here, another arginine on top, a lysine on the side, and a few other sort of hydrophobic amino acids that really tuck away a phosphotyrosine. So when a phosphotyrosine containing protein sort of floats by an SH2 domain containing protein, the SH2 domain can bind tightly to that by coordinating the negative charges on the phosphate using its arginines, and also by sandwiching this phenyl ring between two positive charges in what's called the cation-pi interaction. So we have basically protein domains that can specifically recognize the phosphorylated forms of amino acids. Why would something like this be useful? Well, from a signaling standpoint, this is particularly useful for bringing proteins together in a switch-like manner. So, for example, imagine you have a protein, which I'm just drawing here as a long squiggle tethered to a cell membrane. When it's unphosphorylated, it's doing its own thing, nothing's interacting with it. Once it becomes phosphorylated, SH2 domain containing proteins can anchor onto it. And if these SH2 domains are tethered to some other domain, they can bring other things along for the ride. And in doing so, co-localize proteins that may have some sort of role to play together in signaling. And importantly, this co-localization is dependent on phosphorylation. So it's regulatable and it's switchable. So this is just one way of saying that phosphorylation provides docking sites on proteins for other proteins to bind. But in fact, phosphorylation can also alter the structure of the protein to which it, on which it happens. What's pretty neat about protein phosphorylation is that one of the most common substrates of protein kinases are actually protein kinases. Every single protein kinase, which roughly adopts this fold, which I'll talk you through in just a second, is phosphorylated on a common motif. It's basically this pink motif here called an activation loop. So all protein kinases have roughly the same structure of their enzymatic domains. There are these two lobes. They kind of look like a little Pac-Man. And in between these lobes sits a cleft where ATP binds. I'll show you what that looks like in just a second. And in front of this cleft, basically substrates can dock. And what's really amazing is that when protein kinases are phosphorylated on this loop, you'll see this little cycle through again. It basically opens up this wedge, allows ATP to bind, and extends this pink loop so that substrates can dock. I'll show you pictures of substrates docked a little later in the presentation. But this is just to say that this switch, this turning on of the kinase, opening up of the kinase, is dependent on phosphorylation. So let's see if I can get that movie to run again. This tyrosine on the activation loop gets phosphorylated. It coordinates with arginine, stabling this structure into a basically active conformation. And so all protein kinases can be phosphorylated on these activation loops. And so basically all protein kinases can be turned on by phosphorylation, which alters their structure. This is a relatively subtle structural change, just a sort of unraveling of a small loop and a sort of hinge-like motion of the single domain. But in fact, proteins that have multiple domains that undergo phosphorylation can also undergo conformational changes, sometimes quite enormous ones. So here's another example of a protein. Here's one. It's also a protein kinase, as it turns out. It has a couple of domains tethered to it. One of them happens to be one of these SH2 domains that I mentioned a couple of slides ago, which can bind phosphotyrosine. So here's a kinase that exists in this open conformation with its three domains kind of splayed out and just flopping around. But when this short tail, which is tucked away here, when this short tail gets phosphorylated by another kinase, the SH2 domain, which is tethered to this kinase, comes in and forms an intramolecular interaction, basically stapling down this kinase into this sort of ball-like globular structure. And in this particular conformation, the active site becomes blocked through a sort of structural mechanism I won't go into in detail here. But this is just to say that protein phosphorylation can dramatically alter the structure of that protein in a way that impacts enzymatic activity and, as a result, cell signaling. So I want to talk very briefly about a particular research topic that I find interesting within the realm of protein phosphorylation. And this topic stems from this really simple observation. When the human genome was finally sequenced, I guess now it's been a couple of decades since this happened, we realized that there's over 500 different proteins encoded in our genome that are protein kinases. And so this is just sort of like a cartoon depiction of an evolutionary tree showing the relationships between different kinases. And it's not too important what this diagram really means, except to say that there are basically six or seven major categories of protein kinases, and they're all pretty large families. But what's important is that every single one of these protein kinases catalyzes the exact same biochemical reaction. They take ATP, they remove the distal phosphoryl group, and put it onto the hydroxyl group of a protein on one of these three side chains. And all the structures of these different protein kinases look pretty much the same. So if you have 500 enzymes running around catalyzing the same biochemical reaction, and they all kind of look the same, how on earth does signaling happen with any fidelity? How is there not just a scrambling of information in the cell? And so, of course, these kinases, each one of these 500 kinases, must have some way, or encoded within them, some degree of specificity. They must have a way of selecting some fraction of the proteins in the cell and dismissing the other ones that they're not supposed to work on. In other words, these enzymes must all be specialized in some way. So to understand how specificity is encoded within signaling pathways, basically, I think the consensus is that there's at least three layers in which this happens. So at the biggest layer, it's sort of obvious. If you're a multicellular organism like an animal, if you have a kinase that's expressed in your liver cell and some substrate that's only expressed in your brain, you can bet that that kinase and substrate are not going to be a sort of enzyme substrate pair. But they're never going to see each other, they're not expressed in the same cell type. So this is an important layer of controlling signaling specificity, but it's kind of boring from a molecular standpoint. Then, as we've already alluded to through a couple of slides that I presented and some that Professor Kirschenbaum did, proteins are localized to different compartments in the cell or different regions of the cell, and only those proteins that are co-localized can be enzyme-substrate pairs, of course. If an enzyme and a potential substrate never see each other, same with the cellular level, the tissue-specific expression point, even within the cell, they never see each other, there's never going to be a phosphorylation reaction. And then when you finally get down to the molecular level and you look at an individual enzyme, right, these are proteins. They have sort of fine features on their surfaces, and different proteins, through subtle changes in their structure and sequence, can actually undergo distinct molecular recognition events. So one protein might be able to recognize a particular sequence, amino acid sequence. Another related protein that's slightly different might be able to recognize a different amino acid sequence. So I call this substrate specificity, basically. And this is the topic that I've been studying quite extensively for a couple of years in the context of kinases and now in the context of phosphatases as well. So I just want to illustrate to you how one might actually conduct research in this field and what sorts of findings you might actually gain or discover. So here's another cartoon structure diagram of a protein kinase, this time with a substrate dot, and the substrate is shown in this sort of tan color with the tyrosine shown in pink. And so if you look in detail at this structure, you'll actually find that there's sort of like a fuzzy lock and key. There are parts of the substrate. I've only highlighted two of the residues in this substrate, but they might be able to form very specific types of interactions with the surface of the kinase. And another kinase may have a slightly different surface and thus phosphorylate different substrates. Here are some actual biochemical data showing two kinases that are very closely related to each other. They're very similar kinases. But one of them, this blue one called LCK, will phosphorylate these three sequences, and the green one called CSARC will phosphorylate these three or four. But they're not identical. This is just to say that kinases have some sequence specificity. So when I started generating biochemical data like this, I immediately realized that if you really want to understand kinase substrate pairing and recognition, you need to characterize thousands and thousands of kinase substrate pairs in order to really understand this. And so without delving into the details, I'll just say that I developed an approach where you could take you could take your favorite kinase and you can basically ask how efficiently it'll phosphorylate 3,000 different sequences, or 10,000, however many you want to do, but in this case it was 3,000. And I did this for different kinases, in particular 7 kinases that are just listed here. The names aren't particularly important. But what these graphs are showing you, each dot corresponds to a sequence, a substrate sequence that might be phosphorylated by a kinase. And its position along the x-axis is how quickly it was phosphorylated by this kinase CSARC. And its position on the y-axis is how quickly it's phosphorylated by whatever kinase I'm comparing it to. And so if I just compare SARC to SARC, basically I just do 2 experiments that are identical, you get roughly a line saying that the experiment is pretty reproducible. If you compare SARC to another kinase called FIN, you start to see some outliers. The line becomes a little bit fuzzier. And as you go to more and more distantly related kinases, you find that the correlation totally breaks down. What this means is that As you get further away in sort of evolutionary relationships between kinases, the kinases have diverged in what sequences they will recognize in their active site. So this was a finding that emerged from this, and in some ways it's not that surprising. Where the analysis became really interesting is when we started to actually look at what sequences were robustly phosphorylated by each of these kinases. And so I'll just highlight two of them here, or two sort of averages of sequences. One for this protein called LCK. So what these graphs are showing is I basically just ranked the thousands of peptides and said what features are found in the highly phosphorylated substrates. Those are above the line. So for LCK, next to the tyrosine, there's preference for hydrophobic amino acids, isoleucine, leucine, and valine. And it tends to not prefer, so things below the line are disfavored, things like glycine and serine. So that's one kinase. There's another kinase called ZEP70 that has just completely different preferences. It just wants negative charge all over its substrates, E's and D's everywhere. And what makes comparisons like this actually interesting is that these two kinases are expressed in the same cell. They're found in your immune cells, and it's critical that there's no crosstalk between their activities. One kinase is supposed to do one thing, there's a division of labor, so the other kinase is hardwired to do something else. And if they were to crosstalk, in fact, we can do a cellular experiment and show that if you engineer the kinase to crosstalk with the other one, the cell just gets kind of messed up. It doesn't function the way it's supposed to. So this kind of orthogonality is really critical in kinase signaling pathways. You can do the same sort of analysis by comparing other kinases. There's a receptor kinase called epidermal growth factor receptor that coordinates its activity with two other kinases called SARC and ABL, and it's important that these kinases have distinct substrate preferences, and in fact they do. That's all I want to say about these sorts of measurements, but I'll also point out that we can then take these sorts of analyses, these sort of maps of sequence features, and try to understand how these sequence features are actually engendered upon the kinase, or the sequence preferences are engendered upon the kinase, through an analysis of protein structures. And so here I'm just zooming in on the active sites of LCK and ZEP70, basically what I can tell you is that through structural analysis, both experimental and computational, we can get some hints as to why LCK likes hydrophobic amino acids and why ZEP70 likes negatively charged amino acids. Okay, in the last 2 or 3 minutes, I'll just shift away from my own research and tie this back to something about our basic understanding of signaling biology. So you guys have been talking, it sounds like throughout the class, a little bit about cellular signal transduction, or cell signaling, biosignaling as it's sometimes called. We tend to think of this process, this sort of general concept as being described by processes where somehow a cell detects an extracellular stimulus, say a hormone binds to a receptor, for example, and that binding conveys information into the cell and turns on, say, an enzymatic cascade. For example, this cascade of kinase reactions that Professor Kirschbaum already showed in the slides, or it can turn on the production of a second messenger, as you guys also discussed, or just the assembly of protein-protein interactions. But in every case, cell signaling pathways, we typically find that when they're, what their purpose is to basically convert an extracellular stimulus into a transcriptional response. So at some point, the signal makes its way into the nucleus, and it alters what genes are being expressed, and thus what the cell is able to do, or what it chooses to do. Of course, different cell signaling pathways are going on all at once in the same cell. It's not just one pathway per cell. And so you have often parallel pathways, and I'm just cartoon drawing these generic pathways here. What's quite interesting about cell signaling is that when you look at diseases, oftentimes it's signaling proteins that are somehow defunct. They're either overactivated or they're just turned off through mutations. And the most common sort of set of diseases where this occurs are cancers. So, for example, let's say you have one pathway that's supposed to turn on cell growth and proliferation. What frequently happens in cancers is that one of the enzymes in that pathway will become mutated so that it's perpetually on. It no longer cares what signal is coming from the upstream receptor. Whether the receptor is being stimulated or not, the enzyme is just on. We call that constitutively active. And so you often get a constantly on cellular response, or sometimes if the enzyme is hyperactive, you get an augmented cellular response. So this is the way we typically think about oncogenes or kinases or enzymes in general that will transform a cell into a cancer cell. In fact, we're now starting to realize from studies of enzyme specificity, such as kinase specificity, that what can also happen is So you can have an enzyme which is not involved in the signaling pathway for proliferation or growth that can basically, through mutation, undergo a switch or an expansion in what substrates it'll phosphorylate and now tap into a pathway it's not supposed to be touching. So this is a sort of more newly appreciated alternative mechanism that's being found in cancer, in particular with respect to protein kinases. And I'll just end this topic by pointing out further the biomedical relevance of studying protein phosphorylation. So now we've not only sequenced one human genome, but we've sequenced thousands of human genomes, many of which are derived from cancer patients. And what we've learned is that very frequently kinases and phosphatases are mutated in cancers. And so as a consequence of this observation, there's been this enormous push to characterize the structures and the functions of numerous kinases. Here's this one pathway that's frequently mutated in cancers. This is one where a receptor, tyrosine kinase, that same epidermal growth factor receptor that I told you about, is mutated. But we also frequently see other proteins, like this lipid kinase and phosphatase are mutated. This serine kinase gets mutated. Another serine kinase gets mutated. The point being that basically kinases, which are driving signaling pathways through phosphorylation, are frequently mutated in cancers. And because of this, there's also been this huge push to develop drugs that will actually inhibit the activities of those kinases. And so, there's been this sort of wave in the cancer treatment field to develop targeted therapies. When you recognize that a particular kinase is driving a cancer, you want to develop a small molecule or some other kind of molecule that can selectively turn off the activity of that kinase. So hopefully this gives you some appreciation for the sort of structural basis of signaling by phosphorylation, as well as the types of research you can do in this field, and the implications of phosphorylation for studying diseases like cancer. I'll take any questions.
Thanks very much, Neil. Well, I think we can kind of have a little bit of a discussion if students are interested. I really appreciate this topic coming up right now for a couple of reasons, because it actually is a nice segue into the topic we'll be covering at the very end of this class, which is control of gene expression. And as Neil mentioned, very often or almost invariably, these signals end up with some kind of transcriptional response. And so we'll see how that plays out in altered gene expression over the course of the next couple of lectures. And the other reason is because what this subject brings up is actually related to the very first lecture that I gave at the beginning of the semester, which is increasingly therapeutics are being targeted for individual patients. And you may remember that I showed you a biopsy from a patient where they were trying to understand whether or not a particular growth was associated with an aggressive cancer state, and they were taking a look among the data that I showed you, was looking for mutations in AKT. And I said, you might not know what that is right now, but as your understanding of biochemistry improves over the course of the semester, you'll be able to appreciate why a mutation in an enzyme like AKT may result in a disease state like cancer. So with that, are there any questions for Professor Shaw? Yeah.
You mentioned that SH2 domains can bring other things along for the ride. Could you give an example of that?
Yeah. So more often than not, SH2 domains are tethered to some kind of catalytic domain. So for example, I showed you an example of an SH2 domain that's actually attached to a kinase domain. So one example related to the work that I do, I mentioned one of these kinases was called ZAP70. So what this kinase does, it actually has two SH2 domains followed by a kinase domain. So there's this three-domain structure. And these three domains, they basically ball up upon one another and stay, and they lock the kinase in an off conformation. And this globular chunk is just floating around the cell, basically doing nothing. The SH2 domains are distorted, the kinase domains are distorted, but this thing is super stable. So this enzyme exists in the class of immune cells called T cells, which have a receptor that detects basically other unhealthy cells. When that receptor is engaged, it gets phosphorylated on the inside of the on the intracellular chains. So now you've got all these pairs of phosphorylation modifications on the inside of this receptor that are like perfectly spaced for the two SH2 domains of ZAP70. So ZAP70 gets concentrated at the membrane where this receptor is, and in the process basically unravels and unleashes its kinase activity. So this what I just told you, basically it's the same thing that you'll see over and over again when an SH2 domain is attached to a kinase, a phosphatase, or some other kind of enzyme. I will also mention that there are some SH2 domain containing proteins that are literally just adapters. They just have like one or two SH2 domains, and then maybe one other domain that binds some other kind of modification or some other kind of chemical entity, and they'll just tether different proteins together that have these different features. Does that make sense? Yeah.
Did people in the back hear the question? No. Okay. So the question was, to what extent is expression of kinases cell type specific, and can we express one kinase in a different kind of cell type?
So to answer your first question, I don't have a sort of numerical answer, but I don't believe there's any cell type where all 500 kinases are expressed. Within within each there are 100 of the 500 kinases specifically phosphorylate tyrosine. Those are the tyrosine kinases, and those can be further divided into families of very closely related proteins. They have the same domain architecture, basically. And usually what you'll find is, let's say there's 10 proteins in this subfamily, you'll never find more than one or two of those in the same cell type. And so every cell in your body probably isn't expressing more than 100 different types of kinases, but that's a bit of an arbitrary number, but it's certainly nowhere near 500. And your second question was, can you express one of these kinases in a cell type and affect a change in function? And that's definitely true. In fact, one of the ways that people have shown that, say, two kinases in one of these subfamilies, they're actually distinct in their function, is that you'll take one that's expressed in this cell type, transplant it in the other one, and you'll see that the cell no longer functions the way it's supposed to.
Other questions?
When you were talking about cancer cells and inhibiting enzyme activity so as not to have tumor growth, how specific is that? Will it inhibit all of those enzymes, all of those types of enzymes? How does the body function with the drug in that whole system?
So the question is basically, effectively, how specific are these targeted therapies for cancer cells, if I understood correctly? And so it's definitely the case that, all of your cells will have this NAP kinase called ERK, for example. Just one example of a kinase that's being targeted by drugs. And the presumption with many targeted therapies is simply that this kinase is sort of hyperactive in the cancer cells. And so if you hit the right dosage, you can sort of suppress that hyperactivity. But there's another way to look at it. It's not just about hyperactivity, but sometimes what happens is these kinases, the reason they're hyperactive is because they're mutated in a way to make them distinct from the version of that enzyme that exists in all your healthy cells. And so in those cases, there's at least the potential to design a drug that will only target that mutant form. And that's not really something that we've been able to achieve in most cases yet, but I'd say that that's where people's interests are heading.
Sorry, kind of going off that, if there's like just such variety of the amount of mutations that a kinase should undergo that would cause it to be hyperactive, how can we create like clinically targeted solutions since it would be different for each patient?
That's a great, yeah.
That was my next question. Thank you.
Yeah, so it's true that for any given kinase, you may find many different types of mutations that will activate it, right? And so then the question is, how do you design some kind of universal therapy that would work for any one of these? And so that's the reason why, that's part of the reason why thus far, the focus has really just been on inhibiting the kinase no matter what it looks like, and taking advantage of the fact that the kinase is hyperactive in cancer cells and not in others. The alternative, of course, would be that if we really understand in molecular detail what each of these mutations is doing, perhaps we could design a therapy for each, but I think that we haven't reached that level of understanding of structure complemented with drug design.
I mean, that's a real emerging topic in molecular pharmacology. We'd love to be able to do a better job of that.
Have you developed a method for inhibiting non-receptor tyrosine kinases?
For inhibiting non-receptor tyrosine kinases? So there are a lot of like if you when you say inhibiting, you mean by like drug-like molecules or something? Yeah. So there are actually quite a number of existing compounds that people have been developing for the past 3 decades, really, that will target different subsets of protein kinases, whether they're non-receptor tyrosine kinases, receptor tyrosine kinases, or serine or threonine kinases. I personally haven't been involved in the development of any of these drugs. I've been mostly focused on understanding just how these proteins work and what they do in a test tube and in a cell. But for certain sets of non-receptor tyrosine kinases, there are in fact clinically approved drugs already available.
Some of the stories about development of these drugs are absolutely fascinating and like really gripping from the scientific standpoint, and they've also made a huge impact in the clinic. So there's one story about the molecule Gleevec, and I'll try to post a video about one of the scientists who was responsible for helping the discovery of that particular drug and its effect on patients suffering from leukemia.
Do you think like the route of using kinase inhibitors to treat cancer will be more effective overall than immunotherapy?
That's a very interesting question. So yeah, I think at this point where cancer treatment has gone is that there's sort of 2 schools of thought, which I should point out are not mutually exclusive in their actions. So there's cancer immunotherapies where you harness our natural immune system's ability to fight cancer cells and maybe augment it. And the other would be to develop targeted therapies that hit, for example, kinases or other specific signaling enzymes. So what people have basically where these 2 fields are right now is that the cancer immunotherapy field, when they have success, it's a home run. But it's still difficult to get the vast majority of patients to respond to different cancer immunotherapies. There's a few clinically approved immunotherapies, and they don't work on like 90% of the patients that have a particular kind of cancer. They work on 15 to 20%, except in a few cases. There's something called, we can talk about that afterwards. There's a few cases where there have been real home runs for all patients. So there's this issue of like hitting a large swath of the population and also keeping costs reasonable. Cancer immunotherapies are extremely expensive. The flip side then is, okay, what about these targeted therapies? The caveat to targeted therapies is that invariably when you inhibit a kinase, for example, with a small molecule, because of the nature of cancer and the fact that cells are constantly mutating their genome in cancers, there's a fair chance that you'll develop resistance to the targeted therapy. So what some of the pioneers of these 2 fields are now suggesting is that the way forward is to use targeted therapies to dramatically reduce the number of cancer cells you have in your body. They're likely to work on a large number of patients, and then for a lasting effect, use immunotherapies. This is something that's been proposed by a few leaders in the field.
Have they ever talked about using them concurrently or not?
Yeah. I don't know that there's any like active clinical trials to do that, but I wouldn't be surprised if it's on the horizon.
I also just want to take a little bit of time to talk about Professor Shah's kind of training in professional development. I was just chatting with him before, and I was asking him about his experience taking biochemistry, and he told me that he never took a biochemistry course as an undergraduate here. That's kind of true. He took my graduate level biochemistry course, and I think that was a lot of fun. I'll just say, for those of you who are interested in these sorts of topics, I encourage you to consider taking a graduate level course. It might be kind of intimidating, but routinely we find that the undergraduates in these graduate courses are at the very top of the grades. They get the top grades in the class almost all the time. So don't be concerned that it's going to slam your GPA. And we're able to pursue some of these emerging topics in really great detail, so I'll encourage you to do that. The other thing that I'll just point out is that, as faculty members, we're here to try to motivate and inspire our students, but very often it's reciprocal, right? And we're inspired by our students, and I was certainly inspired by the research that Professor Shah did in my lab. And I was just reminded about this a couple of days ago. I had a conversation with a pharmaceutical company who's interested in one of the molecules that have come out of our lab, and they were asking me about one particular synthetic step, and they were saying, that must have been really tough to do. Have you had a lot of problems doing it? And I said, no, actually, we introduced that step and reported on it in a paper that was published by an NYU undergrad, and then I realized that I was talking about a reaction that Neil had conducted for the first time. So we really benefit from your contributions in the lab, and I will take this opportunity in front of other undergrads to thank Neil for his contributions to our research effort. Are there any? Are there any quick questions for Neil about his professional development or training? We could take those now, or you could just maybe pester him right after lecture's over. All right. Well, let's talk about some other post-translational modifications. Thanks again.
Thanks for having me.
Okay, so indeed, phosphorylation is not the only post-translational modification. The cell needs to do a lot to kind of clean up or alter the functional groups that are present on proteins. We saw quite a bit of detail about how RNAs are processed in the cell. What ends up being generated as an initial transcript is not what the cell finally needs. So that processing is important, and very often protein structures are processed in somewhat similar ways. We talked about the fact that the initiator codon for protein synthesis is always N-formal methionine in prokaryotes, methionine in eukaryotes. Okay, well, that makes sense, right? We've got AUG, that's the start codon message, and that's the amino acid associated with that codon. But we don't always need or want that amino acid there, and so very often that N residue position is just cleaved off. Sometimes we don't want a positive charge site at the N-terminus of our protein, and so very often we also see that charge removed by the acetylation of that N-terminal primary amine. That's most of the processing that occurs at the terminus. We do occasionally see some modifications at the C-terminus of a protein, but that's much less common. As far as cleaning up or processing protein structures, we oftentimes see that there is a signal element associated with the N-terminus of a protein that provides some kind of address, a postal code, if you will, that tells the cell what to do with it and where that protein needs to go. And once the protein is directed towards the right place, it no longer needs that address, and that signal sequence is removed. So these N-terminal bits on the protein really determine the subcellular localization. And that often tells the ribosome where to make the protein. So here's a ribosome sitting in the cytosol, and it just makes the protein, and that can be directed towards the mitochondria of the nucleus, for example. But in the presence of a proper signal sequence, the ribosome actually ends up at the membrane of the endoplasmic reticulum. And there is a protein complex that sits in that membrane, in the ER, called the translocon. And so that signal tells the ribosome, okay, begin the process of synthesizing that polypeptide associated with that translocon, and that ends up telling the ER, yeah, okay, we can direct that protein to a secretory vessel or actually direct it for extracellular export, or insert that protein within the plasma membrane. So those signal sequences need to be processed. They need to be removed by that translocon and the signal peptidase associated with that. And so we oftentimes see an N-terminal sequence, a hydrophobic segment following very shortly after that, and then a few more amino acids, and then a site where the protease can cleave that signal sequence off. And you'll see that these sequences really vary quite a lot from one to the other. So these are all proteins that when they're synthesized have this signal sequence associated with it, and that signal sequence gets cleaved off right at that position. So yeah, those signal sequences direct the proteins for synthesis at the endoplasmic reticulum. There is an entity called the signal recognition particle, which plays a really crucial role in that. I'm showing this figure because it nicely shows the mRNA and the fact that this is a cycle that happens with the ribosome associating with that mRNA and then getting directed to the endoplasmic reticulum, and then the ribosome can get recycled. Here's the image that comes from your textbook, and this does a nicer job of showing the details about how the signal recognition particle actually actively recognizes that signal peptide. There is a docking process that happens with a receptor that sits in the endoplasmic reticulum membrane that's looking for that signal recognition particle. That docking happens. This is actually a GTP dependent process. And the hydrolysis of GTP provides the energy to begin directing the growing polypeptide chain as it's coming out of the exit tunnel of the ribosome. You may remember I showed you the exit tunnel last time when we were talking about the ribosome structure, and it directs what's coming out of that exit tunnel into this channel in this translocon. And then the translocon gets popped open, allowing the polypeptide to transit across that endoplasmic reticulum membrane. Once it gets onto the other side, there can be a cleavage event conducted by a peptidase that nicely removes that signal peptide. We no longer need it. And then as the ribosome continues to crank on that mRNA message, generating more and more of the polypeptide chain, the polypeptide can fold up on the other side of the ER in the lumen. Questions about that? Great. So there's yeah.
Do you ever have a scenario where that destination part of the polypeptide, which determines where that polypeptide is going to be carried within a cell, is basically a mistake is made and it gets sent to a part of the cell that it's not meant for? Is there some kind of checking the mechanism for that situation?
I'm not familiar with any pathological process in which that signal is misread, but I can tell you that you can insert a signal peptide sequence onto the N-terminus of a protein that normally wouldn't undergo this process, and you can trick the cell into sending a polypeptide, a growing polypeptide chain to the translocon that otherwise wouldn't go there. All right. So there's, as this image implies, there are in fact a lot of post-translational modifications that happen as part of this process, as part of a polypeptide chain being generated at the ER and being directed into the lumen of the endoplasmic reticulum. And very often, those include the addition of a lipid group onto that protein chain. Okay. Does anybody remember, we talked about this before, why lipid chains are often inserted onto a protein? What ends up happening to those proteins after a lipid chain is placed on there, like a farnesyl group or a geranyl group? Yeah. Right, exactly. So these allow for the association of those proteins with the membrane, right? So these are long hydrophobic alkyl chains. Sometimes we call these lipid anchors, right? They don't want to be in the cytosol, in the aqueous environment. These are super hydrophobic linear chains. Where they want to reside is in the membrane itself. And so, yeah, if we covalently associate these lipid groups with the protein, that's going to be just like an anchor, right? It will keep the protein tethered right onto the membrane where these things are inserted. So the different types of lipid groups, like these prenyl groups or a myristoyl group, there are specialized enzyme families that are responsible for conjugating these things, oftentimes to specific amino acid side chains on the protein. So these palmitoyl groups, for example, are often conjugated onto a cysteine thiol by a particular class of transferase enzymes. Okay, so talked about removal of the signal sequences. There's other types of proteolytic processing that often happens to proteins as part of the general description of post-translational modifications. And we actually talked about this a little bit with regard to active versus inactive enzymes, and we see these very often come up for proteases. Right? We want to make a protease within the cell. We don't want it necessarily to begin immediately chewing up everything in sight. That might be kind of deleterious. So we make these things initially in an inactive form and then trigger them to become active precisely by clipping these enzymes and removing a part of the chain. The inactive form of the enzymes are called zymogen, and they can be then converted to active enzymes. So both chymotrypsin and trypsin are initially generated in inactive forms called chymotrypsinogen and trypsinogen, and then processed and turned into their active form. The processing of chymotrypsin is, perhaps particularly complex. There's two peptide regions that are actually completely removed. Serine 14, arginine 15 are cut out, and another dipeptide at positions 147, 148 is also completely removed. And so this takes the chymotrypsinogen precursor and chops it up into 3 different polypeptides. It's at that point that it becomes active itself as a protease. So what do you think keeps that protein structure together after it's been chopped into 3? Do you have an idea? Yeah.
Is it sort of like a catalytic triad where they have some interaction in that same way?
Yeah, interesting idea. We'll actually take a look at a catalytic triad in a protease in just a second. That's not what holds these chains together, however. Other ideas? Disulfide bonds. So as these things fold up in the inactive state, it positions disulfides in proximity. They form the disulfide linkage. That's a non-local covalent association that can then be used to hold what would otherwise be separate polypeptide components together in 3-dimensional space. There are other proteins that require quite a bit of cleavage. Collagen is a structural protein that requires proteolytic processing to become mature, and I'll show you an example of some hormones that also need maturing. Your blood clotting sequence, there's a whole cascade of proteolytic events that happens when you initiate blood clotting, and that requires a lot of zymogens to be converted into their cleaved and active state. So let's take a look at the structure of an enzyme, a protease, in its zymogen inactive form and its processed active form. So we're going to compare plasminogen and plasmin in blue and in red. And we see some particular, here's some disulfide linkages holding these things together. And some different amino acid side chains have been highlighted. Can you guess what the active site of this thing might be? This relates to the question that you brought up before. You pointed out a catalytic triad. Anybody see a catalytic triad here? Go ahead.
Histidine, serine, asparagine?
Ser, his, as, right? So there's our catalytic triad. And it's after cleavage that we really get this positioned very nicely. You can see this huge switch in the position of that tryptophan, which actually looks as though it's also very much associated with the formation of the correct active site. So yeah, these processing events can really influence whether or not an enzyme is active or inactive. Right, so this is not actually showing 3 different bits, and we're taking a look at plasmin, not chymotrypsin. This just shows you that there are disulfides, not so much meant to show you that it's holding up protein chains together. All right, so processing of protein structures, sometimes we generate a huge long polypeptide chain, and then we want to clip bits out and use individual components of those as peptide hormones. And this just shows the proteolytic processing of a huge long hormone precursor, and depending upon whether you're in the pituitary gland or in the nervous tissue, you might find different types of processing of this initial polypeptide chain as it comes off the ribosome to release, let's say, endorphin or say this lipotropin peptide hormone, or inteflin, for example. So these processing events can be different in different tissues. I'm going to talk a little bit about amino acid modifications. I'm going to defer, I guess, a discussion of ubiquitination until next time. I'll just cover this quickly just to say that there are a bunch of different specific chemical modifications that are made onto amino acid side chains in addition to the phosphorylation that we really covered in good detail. Professor Shaw mentioned that we don't very often see 2 negative charges on an amino acid side chain. We can introduce a second carboxylate group onto the side chain of glutamic acid by carboxylation. And when we do that, this actually allows the side chain to complex the divalent cation of calcium. Methylation, we see this on lysine, arginine, or glutamic acid residues. These are carried out by a family of enzymes called methyltransferases. You can see this can be done once, twice, 3 times onto a lysine side chain. Typically, these are irreversible changes. We don't change the charge state of the lysine. We still retain the positive charge. It's a slight change in sterics. You can imagine this has a dramatic change in the hydrogen bonding capability of these side chains, and as a result, it can alter protein-protein interactions, and we'll see how this might play out in variation of histone structures when we talk about gene regulation. Just give you one more example of a post-translational modification of an amino acid side chain, and that is onto this structure of collagen. And we talked about collagen before as being a really important structural protein. It has a typical triple helical structure, and it's got a very characteristic repeating tripeptide sequence of glycine, proline, and hydroxyproline. This is an incredibly common protein in our bodies, and as a result, hydroxyproline is one of the most common amino acids that you'll find in protein structures. But you can look at the genetic code all you want, and you will never find hydroxyproline encoded by any codon, and it's never brought to the ribosome and incorporated into a polypeptide chain. Instead, the cell makes immature versions of procollagen, and then at each one of these proline positions, there's an enzyme that's responsible for inserting a hydroxyl group onto the proline side chain. And that is done by this enzyme, prolyl 4-hydroxylase. So that's just one more example of a post-translational modification onto a protein structure. We'll wrap this all up and talk about how, in fact, whole proteins can be added onto glycine side chains by introduction of ubiquitin groups, and how that targets those proteins for degradation in the cell. Anybody who wants to chat with Professor Shaw or any follow-up questions, feel free to check out. Otherwise, I will see you guys on Thursday.
She's not gonna let me grab
No, Kyle. You know, she doesn't need to let you do that. Grab one study sheet.
Yeah. It was like 3 seconds later.
I wasn't going to the library.
What?
I wasn't really going to the library.
Yo, Kyle, what did you say?
I said I wasn't
Are
going
y'all
to
going
the
to study,
library.
study session?
Not today, no.
Study session. You're going? Are we just going and grabbing and leaving?